Tonight, when you open your medicine cabinet, you probably see a row of ordinary objects. A painkiller box, a bottle of allergy pills, maybe a leftover antibiotic from the last time you got sick. Each one looks simple, like a small promise in plastic or cardboard, but inside those pills is a hidden architecture so intricate that for most of modern history, no one really knew what it looked like. That hidden architecture is made of proteins. Proteins are tiny molecular machines built from chains of amino acids, and they fold into elaborate three-dimensional shapes that decide what they can do. Shape is everything. A small twist can turn a helpful protein into a useless one, or even a dangerous one. Your body is full of these machines, building, repairing, signaling, and defending every second you are alive. How do you design a drug for a machine you cannot see? For decades, biologists knew the recipes. They could read the sequence of amino acids, like letters in a sentence. But turning sequence into shape was a brutal puzzle. Experimental methods like X-ray crystallography and cryo-electron microscopy could reveal structures, yet they were slow, expensive, and often failed. Each solved protein could take months or years of work. By the early 21st century, the world had sequenced hundreds of millions of proteins, but only a tiny fraction had known shapes. Can you imagine trying to fix a city using only floor plans for one building in 1,000? This gap became one of the great unsolved challenges in biology. It even had a formal arena, the Critical Assessment of Protein Structure Prediction, or CASP. Every 2 years since the 1990s, researchers would quietly compete. They tried to guess the three-dimensional shapes of proteins from their amino acid sequences alone, and independent experimenters would score them. For many rounds in the early 2000s and early 2010s, progress was slow and incremental. Better than random, but nowhere near precise enough to redesign medicine. Then the century's other revolution arrived, modern deep learning. You already feel deep learning when your phone recognizes your face, when translation apps turn one language into another, when recommendation systems decide what you see online. These systems learn patterns from vast amounts of data, the way your brain slowly learns what a cat or a car looks like. In the 2010s, some researchers began to ask a strange question. If an algorithm can learn to see objects in photos, could it also learn to see shapes hidden inside protein sequences? After all, both are patterns, just in different dimensions. What happens when you aim the same mathematics at the machinery of life? In 2018, during the 13th CASP experiment, a team from DeepMind, led by scientists like Demis Hassabis, John Jumper, and Andrew Sr., entered the competition with a system called AlphaFold. It was not magic. It was a very large, carefully trained deep learning model that treated protein sequences a bit like language and images at the same time. It learned from the known structures that earlier generations of scientists had painfully worked out in labs. When the results were revealed, AlphaFold's predictions clearly outperformed other methods. It was a shock to the field. For the first time, an algorithm looked like it might be genuinely seeing how proteins fold rather than just guessing. But this was only the beginning. Over the next 2 years, the system was rebuilt, refined, and scaled. In 2020, at CASP 14, an improved version often called AlphaFold 2 showed up. Its accuracy on many targets was on par with experimental methods, down to atomic details. One of the CASP organizers, John Molt, described the performance as solving a 50-year-old grand challenge of biology for many proteins. Instead of modest progress, it felt like a phase change. Imagine waking up one morning and discovering that a machine can now sketch the shape of most proteins in your body nearly as well as a physical microscope. How does that change what you assume is possible in medicine? The trick behind AlphaFold 2 was not a single idea, but a stack of them. It combined attention-based neural networks, similar to those that power modern language models, with geometric reasoning about distances and angles. It treated each protein as a kind of flexible graph, then updated its internal picture again and again until the predicted structure became stable. The system was trained on public databases, such as the Protein Data Bank, which stores experimentally determined structures, along with sequence databases curated by teams like those at the European Bioinformatics Institute. It was a collaboration between decades of human effort and 21st-century pattern-finding machinery. You feel the result today not in code, but in how quickly new biological questions can be asked. Soon after AlphaFold 2's performance was announced, another group pushed the concept further. In 2021, David Baker's lab at the University of Washington, long known for protein design, released a model called RoseTTAFold. It also used deep learning to predict structures and was made openly available as software. Suddenly, not just one company but labs around the world could run powerful structure prediction locally. Tools that once existed only in elite crystallography centers began to appear on ordinary compute clusters. When an idea spreads like this, how fast can an entire field shift under your feet? The real impact came when these models were turned outward on the entire known protein universe. In 2021 and 2022, DeepMind and the European Bioinformatics Institute released the AlphaFold protein structure database, containing predicted structures for hundreds of thousands, then millions, and now hundreds of millions of proteins from humans and many other organisms. Overnight, the number of proteins with usable three-dimensional models exploded. It became normal for a biologist studying some obscure enzyme to type its name into a website and see a plausible folded structure in seconds. You might never visit this database directly, but its fingerprints are all over the drugs and therapies that will reach you. Think about what happens when a new disease appears, as it did with the coronavirus pandemic of early 2020. Scientists scramble to understand viral proteins, especially those that latch onto cells or evade your immune system. Experimental structures of the spike protein and other components were crucial, but they took time and intense effort. Now, with predictive models, researchers can generate structural hypotheses early, test which regions might be vulnerable to antibodies, and design molecules that bind more tightly. Even when predictions need refinement, they narrow the search. For a virus that spreads through flights and phones faster than old methods can respond, does cutting months off structural work change the outcome for people like you? Drug discovery fits into this as well. Many medications work by docking into pockets on proteins, blocking or activating them. In the past, if a protein's structure was unknown, chemists often had to guess where the pockets might be, or they had to work with low-resolution information. Now, predicted structures give them a detailed three-dimensional landscape, with grooves, cavities, and flexible loops. Companies like Isomorphic Labs, a spin-out from DeepMind, and many pharmaceutical firms now use these models to prioritize targets, design better candidates, and reduce failures. When a future heart medicine or cancer therapy works on your cells, there is a growing chance its journey passed through an AI's imagined protein model. Enzymes that clean your clothes or process your food are also being reshaped. Industrial biotech companies redesign enzymes to work at higher temperatures, in harsher detergents, or with new substrates. With accurate protein models, they can see which amino acids to swap to widen a pocket or stiffen a region. They can evolve proteins in silico, then test only the most promising variants in the lab. The stain remover that lifts a splash of sauce from your favorite shirt, the bio-based plastic that decomposes more cleanly, the greener chemical process in a factory you never visit. All of these can trace their improvements back to an AI quietly folding proteins in the background. The breakthrough also changes how you think about your own body. Rare genetic diseases often come from a single amino acid change in a protein. Before, seeing how that change warped the structure could take years or might never happen. Now, models can show how a mutation might twist a helix, break a binding site, or destabilize the entire fold. Clinicians and researchers can sort variants of unknown significance by their likely structural damage. They can predict which changes might be rescued by a small molecule or a designed protein partner. When someone in your family gets a confusing genetic report, these tools help doctors move from mysterious letters on a page to a tangible shape that can be reasoned about. Of course, prediction is not the same as truth. Experimental structural biology still matters. There are proteins with multiple shapes, floppy regions, or complex assemblies that stretch the models. Lab measurements confirm, correct, and extend what algorithms propose. But the relationship has flipped. Instead of experiments telling AI what is true in every case, AI narrows the field and experiments focus on what is most ambiguous or important. It is like moving from wandering a dark cave with a candle to walking with a detailed map and a flashlight. You still bump into unexpected corners, but far less often. Do you notice how often in your life maps and navigation apps quietly rearrange what feels possible? There is another, subtler shift. When you think of artificial intelligence, you might think of chatbots, image generators, or software writing code. Protein folding shows AI doing something different, connecting patterns in data to the physical rules of chemistry and physics. The success of AlphaFold and RoseTTAfold hints at a future where similar systems learn the behavior of RNAs, molecular complexes, or entire cells. If a model can learn the language of proteins, what happens when it learns the language of metabolism, the immune system, or brain circuits? The medicines you see today may be only a first draft of what this century will be able to design. All of this runs on the same hardware that powers your everyday apps. Graphics processing units that were built for video games now simulate the contortions of molecular chains. Cloud platforms that host your photos also run the jobs that fold proteins. Research groups in universities log in from laptops not very different from yours and submit jobs to shared clusters. The distance between the device in your hand and the labs redesigning biology has never been smaller. Does it change how you feel about that glowing rectangle when you realize it shares an architecture with machines that model your immune system? The story is also about openness. After the AlphaFold protein structure database launched, millions of predicted structures were released freely. The European Molecular Biology Laboratory and DeepMind made them available to anyone with a browser. Baker's group released RoseTTAfold code. Others built tools like ColabFold so that even small labs could fold proteins without dedicated infrastructure. High school students, small biotech startups, and nonprofit groups all gained access to capabilities that would have looked like science fiction at the start of this century. This kind of democratization shapes which diseases are studied, which crops are improved, which neglected problems finally get attention. You live in a world where supply chains can break, pathogens can emerge suddenly, and climate shifts can rewrite agricultural maps. Many of the solutions people now imagine involve proteins, enzymes that eat plastic, crops with more resilient stress responses, antibodies tuned to new viral variants, microbial communities that pull carbon from the atmosphere. None of these are simple, but they all begin with understanding what a protein looks like and how it moves. With modern AI folding engines, that first step is no longer an impenetrable wall. It is a steep hill, but one with clear paths carved through it. So tonight, when you open your medicine cabinet and see those small, ordinary pills, you can imagine the invisible machines inside them. You can imagine the shapes of receptors on your cells and enzymes in your liver, the docking pockets and flexible loops that decide whether a drug soothes your pain or misses its mark. You can picture teams at DeepMind, at the European Bioinformatics Institute, in David Baker's lab, and in countless pharmaceutical companies, staring at simulated folds on their screens. Their algorithms learn to read the language of proteins so that your 21st century medicine could be written faster, safer, and smarter. Tonight, when you open your medicine cabinet and see a row of ordinary boxes, you will know that behind their simple labels lies an AI that finally taught your species to see how the body's tiniest machines fold into the shapes that heal you. If you liked this video, do like and subscribe to this channel. Comment below on which scientist would you like to watch next.